Let me share the slides first. So in last session we did discuss about fatty acids, by lipids, membranes, and different type of phospholipids, etc. In today's class, we will mainly be discussing about the structures about DNA, RNA, and the different constituents of them. In the last session, I did not discuss about this DNA, RNA structure because as far as I knew that time, your assignment 8 was yet to be submitted, and as the assignments are meant to be solved by yourselves first, then we can discuss the questions. So I did not took up those questions related to DNA and RNAs because I could not officially do that. Okay, and since now the submission has been done and I think the results is also out, so I can discuss those questions if you have any doubt or not regarding that also, so that you can solve this kind of questions in the exams as well, those who are enrolled for the exams. So without further ado, I did not get any confirmation about the whether I am audible or not, but if someone can please confirm that if you can see the slides or not. Okay, just do write in the chat box and that will be very helpful to me. Okay. Okay, thank you very much. So yes, so let us start. Today's questions will be very easy even if you have done all the assignments, and I think Shrija has already discussed this type of question in her doubt clearing session. So it should be very easy for all of you to answer all these questions. You can take it as a form of your exam as well, that if these questions come in your exam, you should be able to answer because Srija has already discussed, ma'am has already discussed, and you have all done the assignments as well. So we will start from question number 1. It states, which of the following process results in the formation of protein from RNA? So these are your 4 options. Today's questions are very easy, to say the truth. So the process for formation of protein from RNA, what do you call it? There are 4 options: transcription, translation, replication, and transmission. So if you do remember, the process that goes on is DNA to RNA to protein. Now in this particular question, they are asking the name of this particular process from RNA to protein. This is your biological dogma. This is the whole process through which entire biology is standing upon. Now you need to tell the name of this particular process. Choose any of the options that are written. A is transcription, B is translation, C is replication, and D is transmission. It is very easy, right? So please do answer. It should be very easy for all of you to answer this. Okay. Since no one is answering yet, let me show you the correct answer. So yes, the correct answer for this should be translation. So you should be very clear about the whole process, that DNA to RNA is known as transcription and RNA to protein is known as translation. You should be very clear about this. This is very easy to mix up between these two names, transcription and translation. Be very clear, DNA to RNA is transcription and RNA to protein is translation. Why it is called translation? Because RNA to protein means you are basically passing on the information or genetic information that which proteins do you require in your body for further functioning. So that information is being translated finally from RNA to protein. That's why that particular process is known as translation. So do not confuse it. DNA to RNA is transcription and RNA to protein is translation, because the informations are being translated. Okay, fine. So this should have been the correct answer for question number 1. Moving to the next one, it is stating which of the following option does not correspond to the pyrimidine bases found in RNA. So there are 4 options. You have to choose carefully. So first of all, they are talking about RNA. So the first important point that you have to note down, that here we are talking about RNA. Fine. We are talking about RNA plus we are talking about the pyrimidine bases. So we are talking about pyrimidine bases. So we all know that there are 2 types of bases that forms these nucleotides. One are called purines and others are called pyrimidines. Now in this particular case, they are asking us to identify the pyrimidine bases of RNA. Please try to answer it. I think all of you know this. I am just giving you for like 20 to 30 seconds. Try to answer it. Otherwise, obviously, I will tell you the answer. I have a query in assignment question. Since let us know how it will once given that answer is correct. Okay. Can you please tell me the type the question here, because the questions are actually handled by the TAs, not by me. There are other TAs in this course who actually handle the assignment questions. So in assignment is question number 9. Let me note it down. What I can do is, okay, question assignment 8, question 9. Okay, so what I can do is I can directly talk to them actually about it. So today I will meet them and let them know about it. If I think they should already know, if this kind something like this has happened, they should know. I will tell them again. As far as I know, if the answer that was given was wrong, mainly what happens is the question is being re-evaluated. That means all of you will be given the mark. And this not always promptly shown on your scorecard for assignments. What happens once the whole course is over, the NPTEL team generally sends us for the assignment evaluation, and if anything wrong has been happened during that, we can rectify after the course has been over. So even if the answer is changed and the marks has been provided to you, it might happen that it will not immediately reflect on your scores, but ultimately it will be credited and it will be given to all of you if the question answer was wrong. Yet I will tell the TS about this problem, and probably they will send you the mail. Okay, okay, fine. Back to this question. So we are talking about RNA's pyrimidine bases. I do not see any answer yet. So as I was talking, that there are 2 types of base, purines and pyrimidines. So what are purines? Purines are A and G and pyrimidines are T, C and U. Adenine and guanine, they are known as purines. Why so? Because they are fused 5 and 6 membered rings. If you look at the structure for adenine and guanine, you will be able to see that purines are fused 5 and 6 membered rings. They are main structure. But in case of pyrimidines, you will only be able to see one 6 membered ring. So purine bases are adenine and guanine, and pyrimidine bases are thymine, cytosine and uracil. Okay, these are the 5 bases and these are your pyrimidine bases. So the only difference between DNA and RNA, if you remember, the only difference between DNA and RNA is that in case of DNA, you have AG and TC. Fine. But in case of RNA, you have A, G, U and C. So the only difference is that in the combination for pyrimidine bases. In case of DNA, you have AGTC, and in case of RNA, you have AGUC. So instead of thymine, you will have your uracil. Okay. So in the question, what should be that? So the question stated, which of the following option does not correspond to the pyrimidine bases of RNA? So it is again stating does not correspond. So we already know that it should have been uracil and cytosine. So first one is uracilin and thymine. Obviously thymine is wrong because you do not find any thymine in case of RNA. So our first option is actually a wrong statement, and in the question they are asking what are the wrong statements. So 1 is a wrong one. Coming to the next one, again thymine and cytosine. Again this is a wrong option because thymine is written here. So this is also a wrong option. Third one is adenine and thymine. Again thymine is written. So 3 is again a wrong option. So all of these are wrong statements. Only correct statements is cytosine and uracil that are present in RNA as pyrimidine bases. Now for the answer, as in the question they are asking does not correspond, that means they are asking for you to identify the false statements. So it should have been option number D, that is 1, 2 and 3 does not correspond to the pyrimidine bases found in case of RNA. Okay. Leaving to that, hope please make it clear that you remember this thing. These are very basics, but you will require this to understand the whole chemistry behind it. Moving to the next one. The third question states that a nucleotide consists of a nitrogen base, a sugar and a phosphate. Second option is a nitrogenous base and a sugar. C is a nitrogenous base, and fourth option is a sugar and a phosphate. So what should be the correct answer for this? You can write your answer in the chat box. I'm constantly reading it. So first of all, they are talking about nucleotide. So if we do remember, how does our basic structure for DNA or RNA looks like? Let me just write it down. And so on, it can go on. Okay. So this is the basic structure for any kind of any DNA or RNA. So what do you have? This is a base molecule written as B. This is your base molecules that either can be A, T, G, C or U. This is your sugar moiety. Either it can be oxy or it can be deoxy. If in this 2' position, this is your 1' position, this is your 2' position, 3', 4', and this is your 5' position. If this 2' position consists of hydrogen, then it will be deoxy. If it consists of oxygen, then it will be oxy. Or in case of RNA, we have the oxy part, and in case of DNA, we have the deoxy part. So ultimately, this 5-membered ring is our sugar moiety. And the last part you can see, this is the phosphate group. Fine. So this is your phosphate group. So how many constituents are there? We have our phosphate, we have our sugar, and we have our nitrogenous base molecule. If these 3 things exist together, then we call them nucleotide. Again, this can be confusing at some times because of the name, but do remember, whenever there are 3 things together, phosphate, sugar and base, all the 3 things are present, then we will call them nucleotide. But if you remove the phosphate part, just this sugar and base part, if you take it out, this will be known as nucleoside. The names are again very confusing, nucleotide and nucleoside. So just remember like this, if there are three things, then it will be nucleotide, and if phosphate is removed, it will be known as nucleoside. So for this question number 3, where it says a nucleotide consists of what? A nucleotide consists of a nitrogenous base, a sugar and a phosphate group all together. So the correct option for question number 3 should have been option A. Okay. Moving to the next one, it says in the synthesis of a nucleic acid polymer, there is a blank thing, dash hydroxyl group of one nucleotide attacks the phosphate of a dash phosphate of a nucleotide triphosphate. So basically it is talking about the polymerization process that goes on in case of forming the phosphate backbone. So in what positions the attacks happen? That is the question. Now let us, for answering this question, we first need to remember how the polymerization process goes on. So generally what happens? So let me draw it again. 3, 4, 5. You have your base moiety here. You have your OH. I'm just talking about the deoxyribose sugar, not talking about any, not talking about the oxy part. It will be same. I'm just talking about the deoxy part. So you have your CH2. This can be attached to a phosphate group and everything. Now here again you will have another nucleotide like this. Say the base is B prime. It can be same base or can be any other base. Out of A, T, G, C, as we are talking about only showing about DNA. So among A, T, G, C, anything can be B or B prime. This one is your H. This is your OH. And here you have your P double bond O minus. O minus and another P double bond O minus O minus. That is what we are talking about, a nucleotide triphosphate. So this is your nucleotide triphosphate. Why we are calling this? It is very obvious from its structure. There are 3 phosphate groups present. That's why we are calling it nucleotide triphosphate. Now you need a polymerization between these two. How does it process? First, what will happen? A base will abstract this hydrogen atom. So now you will have a generation of O minus here that can attack to the, sorry for this line. So that can attack here and it will look something like this. So this is the whole polymerization process that happens. So you have your nucleic acid here which attacks the triphosphate nucleotide here. Now in this question, now you need to identify the positions where the attacks are happening. Let us do the marking. In this case, this is your 1 prime, 2 prime, 3 prime, 4 prime, 5 prime. And in this case as well, it is 1 prime, 2 prime, 3 prime, 4 prime. Sorry, there I missed one CH2 group here. Very sorry for that. It should be attached with the CH2 group. Okay. So this is our 5 prime. And how can we mark these phosphate groups? As this one is immediately attached to the oxygen, that is this CH2O. Immediately after that oxygen, the first phosphate will be called as alpha. Obviously. the second one will be called as beta, and the third phosphate group will be called gamma. Now we have marked all the position, and now we can see from which position to where the attack is happening. So the hydroxyl group for this nucleotide, the hydroxyl group at 3' position is attacking. So this position should be filled with 3', and it attacks what position? The alpha phosphate of a nucleotide triphosphate. There are 3 phosphate, alpha, beta and gamma, but it is attacking at the alpha position. So here the answer should be it attacks the alpha phosphate of a nucleotide triphosphate. So we have done everything. So what should have been? The correct option chosen for this question is option number B. From 3' to alpha phosphate, the attack happens for the whole polymerization process. Okay. Hope this is clear. Moving to the next one. It states, which among the following are not the features of Watson-Crick pairing? So again you have to identify which are not the features. That means you have to identify the false statements that will be given in the options. And in case of Watson-Crick pairing. Okay. So these 4 statements are given. The first one says the permitted hydrogen bonds are adenine with thymine. There are 2 hydrogen bonds, and between guanine and cytosine we have 3 hydrogen bonds. Exactly that what is happened. If you remember the Watson-Crick base pairing, the hydrogen bondings occur between AT and GC. How many hydrogen bonds occur again? Between A and T, 2 hydrogen bonds. And how many occur between G and C? 3 hydrogen bonds. That is exactly stated in the first statement. So our first statement is a true statement, a correct statement. Moving to the next one. It says the Watson-Crick base pair is a non-planar structure. Why does the Watson-Crick base pair happens? If you do remember, the Watson-Crick base pair looks something like this. Suppose this is your one DNA strand and it is the another strand. I'm just showing 2 places. This is how it looks. If it is G and C, there will be 3 hydrogen bonds, and if it is A and T, there will be 2 hydrogen bonds. The exact role of these hydrogen bonds is to put the bases in a planar structure. That is what happens. They are stacked upon one another. That means all the bases that you can see here are parallel. This is one plane, this is another plane. So these hydrogen bonds are actually holding them in this planar structure, for which the everything is happening. Like all the planarity that is there in case of DNA only arises because of this Watson-Crick base pairing, followed by their specific hydrogen bonding, which leads to a planar structure. So the second statement, which states that Watson-Crick base pairing gives rise to a non-planar structure, it is completely wrong. So our second statement is a false statement. Fine. Moving to the next one. Purine and pyrimidine bases are saturated with the hydrogen bonds. So you have your, let me one second, just a general structure if I draw. So let me just draw it here. Just one of the purines I am drawing. This is one of the purines, and if I look at one of the structure for pyrimidines, it looks like something like this: NH2N NH double bond O. So this one is our purine and this one is our pyrimidine. We can see the whole structures for both purine and pyrimidines. There are many double bonds present within the structure. That means these are unsaturated structure. But in the statements, it has been said that purine and pyrimidines are saturated. No, we can clearly see the double bonds. They are unsaturated structure. So our our statement number 3 is also a false statement. Fine. Moving to the last one. The beta glycosidic bond is attached on the same edge of the base pair. What is our beta glycosidic bond? Now we need to know for that. So if you just remember, it is your basic structure for your sugar. You have your OH moiety. Suppose it can be H or OH. You have your CH2OH group here. Here you have your base attached. This particular bond is known as beta glycosidic bond. Obviously, it is bond at the same edge of the base pair. That is what is written here. It is attached with the base, so obviously it will be on the same edge of the base pair. Very obvious, that is from the structure. So again, the fourth statement is obviously a true statement. A beta glycosidic bond is here, which is at the same edge of the base pair. Now let us go back to the first question. We had to identify which were the false statements, that means not the features for Watson-Crick base pairing. So which of the statements we found false? 2 and 3. So our correct option should have been option C. Statement 2 and statement 3 are the non-true statement for Watson-Crick pairing. Hope this is clear. Moving to the next one. It states which of the following statement is not true. That means again you need to identify the false statements. Fine. The first one says the combination of the staircase sugar phosphate backbone and the bonding between the pairs result in a double helix. Is this a true statement or false statement? Sorry. The combination of staircase sugar phosphate backbone. So where does our sugar phosphate backbone lies? If I just draw the structure. So this, on the exterior of the helices, we have our sugar phosphate backbone. And the next one is bonding between the pairs. Where do we have the bonding? In between we have the bonding, right? Both these factors, that is the bending of the sugar phosphate backbone, if you do remember the lecture materials we discussed, because there are single bonds present in the sugar phosphate backbone, so they can freely rotate, and the sugar molecule, that is the 5-membered ring, that can pucker in different ways, which is shown in the lecture material, like in S form or Z form, 2' endo, 3' endo, and different kind of structures like that. And because of this, the rotation of the phosphate bonds, single bonds present in the phosphate structures, and the puckering of the sugars, the bending of these helices occur, as well as to stabilize this conformation. What happens? The base pairing and the hydrogen bonds, all together this leads to a stable double helical structure. Okay. None of them single-handedly can hold the double helical structure. You both need the stacking and hydrogen bonding as well as the sugar phosphate backbone to stabilize the double helix. Okay. So the first statement that is said here, that the combination of both of them are required, is actually a true statement. Okay. Let us move to the next one. It is saying 5 prime to 3 prime strand is known as sense strand, whereas 3 prime to 5 prime strand is known as anti-sense strand. So what does it mean? There are two parallel strands for double helices. I am just writing not in the helical form, I am just writing for your understanding as the normal ladder form. Okay. So one is 5 prime to 3 prime and another is 3 prime to 5 prime, and you will have your bases and hydrogen bondings here. Fine. It is saying the first strand is a sense strand and this is anti-sense. Absolutely correct. The first strand 5 prime to 3 prime is known as sense strand, also known as coding strand. Okay. And the next strand that goes from 3 prime to 5 prime is known as anti-sense strand, also known as template strand. Why it is called so? Because you need replication of DNAs. Okay, for you to survive, you need replication of them. How do you replicate? You start breaking this ladder from in between. You are basically breaking the hydrogen bondings between the bases. And you will have one strand here, another strand here. The bases will sit like this in random manner. I am just writing like that. Here also the complementary bases T, A, C, G, T, A will sit like that. Now you have broken the hydrogen bonds between them. This is your first strand or the coding strand. This is your second strand 3 prime to 5 prime, that is your template strand. Here comes the role of mRNA. What does it do? mRNA comes here. It will take the information from this number 1 strand, that is your 5 prime to 3 prime strand. What information it is taking? That which base pair are supposed here. Why it is taking that information? Because if now you have to replicate, you have to put one strand here and another strand here. Now from one DNA you will be able to get 2 DNAs, right? Now while placing these new strands, obviously you have to make sure that the base pairs are matching, that the complementary base pairing is possible. Exactly complementary strand you will require for that. That information is given to you from the first strand or known as the sense strand or coding strand. Once it knows the code that okay, in this strand this was base pairings were like that, a similar base pairing will be required here to replicate the DNA. So now it will transform this or work as a messenger for this information and give you a new template. That's why, because it gets its information from this first strand, it is known as coding one. And after getting the information, it will just simply place the complementary base pairs here. Using this already present 2 strand as template. That's why number 2 strand or the antisense strand is known as the template strand. Because now mRNA is using this one as template, attaching to it by gaining information from number 1 strand. It is just a casual representation for your understanding. I am just telling it in this sense for better understanding, to easy understanding actually. So that is what happens. That's why we call the first one anti-sense strand and the second one anti-sense or template strand. Fine. So although that was not the question, I thought this should have been discussed. If you would have any doubt in that session, that should have been cleared by now. So our second statement is also true statement. Coming to the third statement, it says the dimensions of the two permitted base pairs are similar. Obviously, if the base pairs, here A is sitting, here suppose T is sitting, complementary base pairs, here G will be sitting, so in complementary C will be sitting there. Now if you need to form a ladder, just think it like that. If you need to form a ladder, the distance between these two strands has to be same. Otherwise, how it will look like a ladder, right? If it does not fit. So obviously, if the distance between AT and GC are not similar, or to say they are not same, you will not be able to get a parallel DNA. That will not be DNA then. That is the basic structure for DNA. The distance is same. So obviously the dimension for the two permitted base pair has to be similar to maintain that structure. So our third statement is true as well. Moving to the next one, fourth option, it says hydrogen bonds occur due to the formation of water spine in a major group. So first of all, there is many things wrong in these statements. How? First of all is hydrogen bonds occur between the base pairs AT or GC. That has nothing to do with the water molecules. It just occur between the base pairs. That is all. And the water spine that is they are talking about. So if it looks something like this. Okay. Suppose it is our minor groove. It is our major groove. It should again be our major groove, and this one should be our minor groove. My drawing is not very good, but this is how the grooves look like in case of double-stranded DNA. Now what they are talking about in the last option or the last statement, that is water spine that grows along the minor groove. That has nothing to do with the major groove. The water molecules or the water spine will grow along the minor grooves like that. Okay. So it has nothing to do with major groove, and also hydrogen bond has nothing to do with the presence of water molecules here. Okay. So the only statement that is false for this case is statement number D. So according to question 7, which was a non-true statement, is option number D. Okay. Hope this is clear. So move to the next one. The rate of hydrolysis is slower in DNA than RNA. Why does this happen? So there are 4 options. First one is saying the ribose sugar present in DNA is in a deoxy form, but it is not so in a RNA. Okay. Second statement is due to the presence of a different pyrimidine in RNA compared to DNA. Okay. Third one is of the stacking of nitrogen bases. And the fourth one says the phosphate in the backbone of RNA favors hydrolysis at a faster rate. So let us, for answering this question, what do we require is to understand the basic structural difference between DNA and RNA. Right? So suppose this is our base structure. For DNA it will be A, T, G or C, and for RNA it will be A, U, G and C. Here will be our phosphate backbone CH2OH, and say it is going on. For RNA as well, we will have our phosphate backbone and this will go on. Fine. DNA, we have our OH group. For RNA also, we have our OH group. The only difference lies is in case of DNA, you will have a hydrogen here, and in case of RNA, you will have a OH here. So now we are talking about the hydrolysis. Where should be the hydrolysis should be faster and why? Because if you do remember, one second. So in case of hydrolysis, generally, just let me write it down. In case of DNA, this strand will further continue what is known as 5' to 3' attachment. Just as I showed the mechanism for polymerization. So after polymerization, it looks something like this, suppose. And this strand goes on. Similarly, here also, in case of RNA, after polymerization or the formation of RNA, it will look something like this. Okay. Now we have a structure. What will happen in case of hydrolysis? How it can hydrolyze? See, in case of RNA, it is possible because you have a OH group at 2' position. How will it happen? A base molecule can easily take up this H plus and then it will generate a O minus. This O minus can attack this phosphate center and leave the rest of the structure that was there downside. This will lead to look something like this O, P, double bond O, O minus, O. So that means the hydrolysis has been occurred, right? But in case of DNA, so this was our DNA and this was our RNA. So in case of our DNA, as there is no OH group present at the 2' position, that means the hydrolysis will not take place easily. If the hydrolysis has to take on, then from outside any base needs to attack here. But internally no attacks will be there. So the hydrolysis for DNA will be much more slower than in case of RNA, where an internal attack can occur and lead to this kind of structure, right? So the reason for higher hydrolysis rate for RNA is due to the ribose sugar present in DNA in deoxy form. Here we do not have OH which can deprotonate, but an oxy form in RNA which can deprotonate and form O minus. So the correct option for question number 8 was option number A. Okay, let us move to the next one. It states a segment of B DNA double helix consists of 101 base pairs. Fine. What would be the approximate length of that segment in nanometer? So in a segment of BDNA, you already know that there are 101 number of base pairs. You have to determine the segment's length. Okay, so these are the 4 options. If any of you can solve this particular mathematical question, please do write your answer in the chat box. Take the help of this table as well. Every information that you require to solve this is given in the table. So we are dealing with BDNA. All the information that you require are given already. Please try to solve it and write the answer. If you can't, then obviously I will be showing you the how to solve it. It will just take like 10 to 20 seconds to solve it. Okay, fine. So let us see how should we solve this question. So what information do we already have? That our number of base pair is 101. Now if you look at B DNA, this particular number means base pair per repeat, or that means base pair per turn. You know that in case of DNAs, if I write it down like this, a coiled structure, then base pair per repeat or base pair per turn means how many bases will be there per turn. Okay, this is one turn, 2, 3, 4 like this. So how many turns will be there? How many turns should be there? First let us identify that. We know there are 10 in case of BDNA. Total 10 turns per repeat. Sorry, 10 base pairs per repeat or 10 base pairs per turn. So let us write it down, 10 base pairs per turn. And altogether we have 101 base pairs. So how many turns should be there? That we can calculate easily. There are 101 base pairs, and in case of BDNA we have to put 10 base pair in one turn. So approximately how many turns would be there in that segment? 101 divided by 10. So that will be approximately 10. It should be 10.1, which we can approximate to 10. So in this particular segments we already know there are 10 turns. Okay. Now we need to calculate the length of that segment. How to correlate again? Now again see, we have another value that is given in this table known as pitch. The pitch is 3.4 nanometer. What does a pitch means? Pitch tells you the length it covers per turn. Say this length. Upon going from this length to this one, upon going from this turn to this one, how much the DNA rises, that gives you the pitch value. Now it says that for one turn it rises around 3.4 nanometer, and we have already deduced that we have 10 turns in our segments. So in one turn it rises 3.4 nanometer. So for 10 turns, how much it should rise? 3.4 into 10. So 34 nanometer approximately. Yes. So our correct option should have been option number A. For BDNA, the rise should have been, or the length of the segment that we are talking about should have been around 34 nanometer. Okay, hope this is clear. Let us move to the next one. It says the circular form of DNA is the result of what type of interactions of the two ends of linear double helix? Sorry. So we are talking about circular form of DNA. I have already showing you the answer. The correct answer should have been covalent. Why so? So the circular form that we are talking about is a tertiary structure of DNA. How does it look like? Circular form like obviously this, but this circle is actually double stranded. Okay. This should have been the structure. It is originally a double stranded. I am not writing the other strand. Okay. It should look something like this. So the circumference of that circle is actually a double stranded helix. Okay. Now they can originally, what structure do we know? We know that the structure of DNA is something like this. But it is not always linear. These tertiary type of structures are taken up by DNA molecules so that it can actually store more, or it can take less place for its storage. Because if you think it like that, ultimately these DNAs are uh, actually storing your genetic information. You will be requiring them throughout your life period. Now, your genetic information, where do you find these DNA molecules generally? You find them, if this is your cell, you generally find them inside your nucleus, right? That means these long chains, if you do remember, after all the polymerization, the length of it can goes up to like 10 centimeter. That is very long. Now, if they are remaining in that linear form, it will be very difficult for a biological system to store all of this in a single cell or all the cells. It will be very difficult. So what happens? Supercoiling starts to occur. Supercoilings or the circular forms, this happens so that ultimately less place is required for storing these DNA molecules. If this is supercoiled now, you can easily store them inside the nucleus. Okay, but if it was in linear form, you would not be able to do it. Just as simple as that. It's like a ball of yarn. If you are just arranging them like a yarn, obviously it will look like a small sphere. But if you start taking the thread from that ball of yarn, then it will be very huge and you will not be able to actually accommodate it. So that's why sometimes that's why actually by covalent interaction, the circular or sometimes supercoiled structures, uh, which are also known as tertiary structures for DNA molecules, occurs, and that happens by a covalent interaction. Just remember that the tertiary structures for circular formation occurs by covalent interaction. Okay, moving to next one. This one is very easy. What should be the complementary strand for this sequence? One sequence is given in this question and you have to identify the complementary strand sequence. Very easy. Let us do that here. So we all know if one strand starts from 3' to 5', another one should start from 5' to 3'. Fine. Next, we have to choose the complementary base pairs. So for A, the complementary base pair is T. For T, it should be A. For G, it is C. For C, it will be G. Again, here is C, so it will be another G. For T, it will be A. For this T, this will also be A. This G, it will be C. And this A, this will be T. So this should be our the complementary strand. Okay. Now let us look at the options and which one matches this. 5' to 3'. Yes, this one T A C G G A C T. Yes, so the only option that matches is option number D. So that was our complementary strand. Okay. Moving to the next question. It states, which of the following is or are true about the stability of DNA? So we have to identify the true statements. That is important, first of all. Let us look at the statements then. The first one says, charged and hydrophilic sugar phosphate backbone are on the interior. What does that mean? We all know that the double helix that we write, we have phosphate backbone here. That means which something looks like this, right? These sugar phosphate backbones are charged molecules because we have our 2 O minus here. Now in the statement it is saying that this charged sugar phosphate backbone stays on the interior. What is the interior side? This side, interior. Will this ever happen? If you have the interiors of the both the strands negatively charged, then will you be able ever be able to keep them close? Because there will be charge repulsion. Already this is a problem in case of this double helical structure. They are stabilized by many things. Now if you want to put the charge inside of the strands, you will never be able to keep close because of the repulsion that will be happening between them. So obviously this is a false statement. Sugar phosphone backbone rise on the exterior of your double helix. It will never stay on the interior of your double helix. So the first statement that is shown here is a false one. Moving to next one. Base pair hydrogen bonding stabilizes more than stacking interaction. Okay. So now we are talking about the interiors. So what do we have on the inside? We have our base pairs. Now two things happen in case of base pairs. One thing is this parallel stacking that I am showing. These bases are on top of each other. There is a stacking. Okay. Like this. They are all stacked on one upon. And the next thing what happens between them is hydrogen bondings. Now it is saying in the statements that the hydrogen bonding are more stabilizing for the structure than the stacking interaction. This is a false statement. Why I am saying so? Because both of them are equally required for stabilizing the two strands. Because in case of stacking, we are having a van der Waals type of interaction. And again, for Watson-Crick base pairing, we are having hydrogen bond interaction. Both of these bonds or both of these interactions are non-covalent. Both of them are equally required for stabilizing the double helical structure. None of them are superior to the other. So we do require both of them for the proper orientation. So the next statement that is again is a false statement where it says that the stacking is less important than hydrogen bonding. This is a false statement. Both hydrogen bonding and stacking are equally required. Moving to the third statement, it says the hydrogen bonding guarantees the complementarity of the strands. How does it? What do we mean by complementary? Complementary means if you have A here, you have to have T. And if you have G here, you need to have C on the other strand. And how this pairing happens? Because of the hydrogen bonding. Because A is stabilized by 2 hydrogen bonds with thymine and G gets stabilized by forming 3 hydrogen bonds with cytosine, that is why they are forming this pair. So absolutely yes, this hydrogen bonding is guaranteeing that they will be sitting in a complementary manner. It will not go to sit here because then it will not have sufficient number of hydrogen bonding that it requires for stabilizing itself. That means only A will sit with T and G will sit with C. So yes, hydrogen bonding does make sure that the complementary basis remains on the other stand. So it is a true statement. And the fourth statement is stacking interactions are sufficient to overcome the electrostatic repulsion of the phosphate. So as I was saying that although the negatively charged phosphate groups are outside of the double helical structure, yet it forms distability, or it provides a distability in the structure, because ultimately you are going to have a coil. So it's like you will have negative charge here, 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 here again. So there are a lot of negative charge which are very near to each other. This is a negative force, so you need to stabilize it by somehow. But just stacking of bases, the van der Waals interaction by stacking of bases is not sufficient to overcome all the negative stabilization that has been happening due to the charges. For that you require both the stacking as well as the hydrogen bonding, just as we discussed. Both are equally important to stabilize the whole structure. So again the fourth option is a false one. Electrostatic repulsion cannot be overcome solely by stacking interaction. You will eventually require hydrogen bonding. So the only true statement for this question is 3. So our correct option should have been option C. 3 only. Okay. Moving to the next one. Okay, fine. There are two gaps. The dash in ribonucleotides renders RNA molecules susceptible to strand cleavage in dash solution. Okay, so we are dealing with RNA molecules. So let us quickly draw the structure. Now I think all of you remember, we have our base group, we have our phosphate group, we have our OH group here, and we have our OH group here as well. Both the 2' and 3' position. Right? Now it is saying the RNA molecules are becoming susceptible for strand cleavage. How it will happen? First of all, for RNA, as I just have said, this will go on to react with another phosphate group and the strand will go on like the 5' 3' linkage. Now to cleave the RNA strand, you will need a generation of O minus here, just as I discussed a few slides back. If there is a generation of O minus, this can attack this phosphate group and then cleave this part from here. So this is, they are talking about ribonucleotides cleavage. So who takes part in the cleavage process? The 2' OH group takes part. So the 1, in place of 1, what should have been? Because already the 3' OH group is blocked in case of RNA. It is already blocked for polymerization. The only free group, OH group, that is there is 2' OH group. So that is going to take part for our cleavage. And in what kind of solution it will be promoted? So ultimately you will require the formation of O minus. So in what condition O minus will form? Obviously in alkaline condition, because the tendency for the attack for O minus is much more greater than OH. So if you have an alkaline solution, then the O minus formation will be much more prompted, and the breaking of this or the cleavage of this strand will also be prompted. That's why the second one should be in alkali. Basic condition, basically more the formation of O minus, more will be the strand cleavage. So what should be the correct option then? The 2' OH in ribonucleotides renders RNA molecules susceptible to strand cleavage in alkali solutions. Okay, fine. Moving to next one. This question states, which among the following statements are false? So we again have to identify the false statements about DNA melting temperature. So we are talking about DNA melting temperature. Fine. The first statement is, the higher the salt, higher will be the melting temperature. What does that mean? let us first draw the double helical structure. So just we discussed that on the exterior of the double helix we have our negatively charged phosphate groups. Fine. Next you want to melt it. Originally they are in a very nicely crystalline state. Now you have to give temperature to take it to a non-crystalline. Okay, 1 second, just give me a minute. 1 second, there is a problem with my phone. Just 1 second, I'll be back. Okay, fine, sorry for that. Just let me go back to it. Where we are talking about, we are talking about this question. Okay, fine, just get back. Okay, so we are here talking about the melting temperatures for DNA. So DNAs are generally highly crystalline structures. Now you are taking it to a non-crystalline state by giving heat from, say, outside. So if I do write down the energy profile, energy profile, it will look something like this. If this is your reaction coordinate and this is your delta G. So DNA was originally very much stabilized. You want to take it to a random state. Obviously you will need to supply some amount of heat to overcome this barrier. Now if this barrier, the delta, if this barrier that is I am showing here is more, that means you will require higher temperature to cross it. If this gap is small, then you will require lesser temperature to cross it. That is all. So this is your crystalline state and this is your non-crystalline state. Okay, this jump you need to take care. So originally suppose it looks like this. Fine. In the next, 1 second, color, let me just change the color. See, suppose in the next part, when you are adding salt in the structure, what is happening? Suppose you are adding Mg2 plus. What will they do? They will sit in between here, between the negative charges. Negative charges were initially repulsing or repelling each other because they are same charge, so initially they are repelling. Now if you are adding a positively charged thing here, that means you are stabilizing this structure actually. No more negative repulsion anymore. You are neutralizing the charge. That means you are stabilizing this structure by adding salt molecules. So obviously your energy now will look something like this, isn't it? Because you have stabilized it by adding some salt. Now after this You are lucky. This one is after adding salt. Now you are trying to move from this state to here. This should have been equal to, there should have been equal anyway. Now look at it. This difference, this delta value is much higher than before adding salt. Why so? Because of adding salt, this state has been stabilized, but this non-crystalline state is same. It is a random state, it is same. So this value, this jump for the transition has been increased. That means you will require more temperature to cross this barrier. So what the salts will do? It will increase the Tm value. So that means higher the salt, higher will be the melting temperature. That should be the case. So our first one should be a true statement. This one. This is our true statement. Next, coming to the next statement. It is saying the higher the GC content, lower the Tm. So what is our GC content? There are 3 hydrogen bonds. You need to remember. In case of A and T, there are 2 double bond, but in case of 2 hydrogen bonds, but in case of G and C, there are 3 hydrogen bonds. Now, if you are increasing your GC content, obviously that means the AT content will be lower, because ultimately the whole thing has to be same. So AT content is lowering and you are increasing GC. That means you are replacing 2 hydrogen bonds with 3 hydrogen bonds. That again means now the structure is much more stronger because there are more number of hydrogen bonds. That means again, if let me choose the thing again, that means again the structure has been stabilized because now we have more number of hydrogen bonds. That means if we need to go to the non-crystalline state, again the delta value will be much more than the original one. That means higher the barrier, higher will be the Tm, or the higher will be the temperature that you will require to take it to a non-crystalline state. That means higher GC contents leads to higher hydrogen bonds, higher number of hydrogen bonds, which increases the Tm value. But in this second statement, it is mentioned that higher the GC contents, lower the Tm. This is wrong, just we saw now. That means second statement is a false statement. Third statement says low and high pH decreases Tm. Obviously, how that happens? What will happen at very low or very high pH values? So let me do it that again. At very high or low pH, the bases that you have, purine and pyrimidine, they can protonate or deprotonate. Any of the things can happen, protonate or deprotonate. So you already know that the bases are involved in hydrogen bonding. Now, due to the pH, if any of them gets protonated or deprotonated, that means their hydrogen bondings will be hampered. That again lead to irregularity in the structure. If the hydrogen bonding is no longer strong, sorry, what is happening? I cannot see the pointer, just give me time. Okay, fine. If the hydrogen bonding is no longer strong, that means your double helical structure is no longer strong again, which implies that the original energy of your double helical structure has increased because it is now non-stabilized. So it is higher in energy. Now if you need to go to the non-crystalline state, the delta that will be requiring is originally less than the crystalline state. This red one was higher, now this yellow one is low. Low barrier means you will require low temperature to cross it. That means Tm will also go low. The third statement says the exact same thing. Lowering the pH or increasing the pH too much will decrease the Tm values. So again, our third statement was true as well. So the only false statement in this case is 2. So the correct option should have been option number B. Okay, fine. Moving to the next one. It states the overall rate of DNA denaturation is determined by what? So what do you mean by DNA renaturation? One second. Let me go back to red. Yes. So suppose we have our DNA like this. We can give drastic condition like very high temperature, very high or very low pH, any of them. These conditions can start to denature the DNA molecules. So basically the hydrogen bondings are broken, something like that. So you are denaturing that. By reversing the condition, sometimes we can go back to the original state. Not always, but sometimes we can go back to the original state. This is called renaturation. So you have already your denatured DNA. Now you are reversing the conditions and going back to a renatured state, and that process is known as renaturation. So how much the renaturation will occur or in what speed the renaturation will occur depends on many things. This question asks about that. The first option is the concentration of DNA. Okay, now you just think it like, suppose you had this bookshelf where many books were stacked nicely. Okay, see, these books are nicely stacked. Now going to random state, now the bookshelf something looks like this. These books are just randomly stacked like this. Okay, it something looks like this. You want to go back to the order state. If the number of books that were in the shelf was more, then you will require more time to rearrange them, right? Because if there are suppose 5 books, it will be very easily rearranged. But if there are 500 books, it will take a longer time, much longer time to rearrange them in the shelf. That is exactly that thing. If the concentration of DNA is more, that means you have more number of books, actually the example, as of the example, that means you will require more time for renaturation, obviously. So obviously the concentration of DNA is a very important factor that they determine the overall rate of DNA renaturation. Okay, so it is an important factor. Checked. The next one is the average length of the DNA segments. Again, same thing. Suppose this was now a small bookshelf. Instead of that, now we are talking about suppose a whole almirah. Okay, where there are so many books that you need to arrange. Now, as the length has increased where you are keeping the books, that means a longer time you will require to arrange them again, because more number or more length means more number of books, that means more time. So again the average length of DNA will affect the overall rate of DNA renaturation. The longer it is, the longer it will take to renaturate. right? So this is also checked. The third option is the complexity of the DNA. Now, up till now, we are just talking about arranging the books. But now suppose I am saying you not only have to arrange 500 books but also have to arrange them alphabetically. Right? Now if you had to do, just you cannot simply put the books again there. That will take again much longer time to arrange them alphabetically. The same things for DNA as well. Because if you increase the complexity in the structure, say there are very randomly complexity means what? Suppose a DNA contains A, T, like that. Okay. But now you are increasing the complexity with something like this. This sequence is much more complex compared to this one. So whenever this becomes very complicated, you will require more time. It's as simple as that. So the complexity of DNA obviously does determine what will be the rate or how much time you will require for DNA denaturation. So for this particular question, as of now, all the three things that we discussed here all are very important for determining the overall rate of DNA denaturation. So the correct option for question number 15 should have been option C, that all 1, 2 and 3 are required or all of them determine the overall rate of DNA renaturation. Okay. Hoping this is clear. Moving to next one. It states, in the central dogma of biology, the process involving the formation of RNA to DNA is called what? So we all know central dogma looks like this: DNA to RNA to protein. And at the very first question, question number 1, we discussed that from DNA to RNA, this is known as transcription, and RNA to protein, this is known as replication. We just discussed in question number 1. The same question was asked here. So our correct option should have been option number B, transcription. Okay. Moving to next one. It says, according to Watson and Crick model, which base pairing is correct? Let us look at that. We already know. In case of G, it base pairs with C and A base pairs with T. So out of all these options, does A pair with G? No. G pairing with C? Yes. C does not pair with T, again wrong. T does not pair with G. So again this is wrong. So the only correct option is option number B. The correct base pairing is G pairing with C. Fine. Moving to the next one. Which of the nitrogenous base is not present in case of RNA? So we know again that DNA contains A, T, G, C. RNA contains A, G, C, U. So instead of thymine, you do replace uracil in case of RNA. So out of that, if you look at the structure of A, B, C, D, then our A is thymine, which is not present in case of RNA. So for this question, that which of the nitrogenous base is not present is RNA, should have been option number A. Okay. So moving to the next one. It says, one of the differences between DNA and RNA is that the type of sugar they consist of. Fine. DNA has a deoxyribose sugar, that's why it is called deoxyribonucleotide, right? From which position in the ribose ring of DNA is the OH group missing? So basically they are asking what is the position, what is the deoxy part where you cut the OH group and place OH. So again Just quickly remember the structure of the pentose sugar for DNA. It is all same. It is your base pair, it is OH. Just in the second position, you have your hydrogen instead of OH. So what is this position? This is our 1, this is 2, this is 3, this is 4, this is 5. So what position? C2. So our correct option should be option number A, that at C2 position the OH group is missing in case of deoxyribose sugar. Fine. Let us move to the next one. Which of the following is true about phosphodiester linkage? So let us see. We already know about phosphodiester linkages. How does it look like? Just let me show it down here. We have our base. We are just considering DNA. And here is another base. We are considering our OH group. Now this was O, P double bond O, O minus, and now we have a link here. Okay, so what is this linkage? 1, 2, 3, 4, 5. 1 prime, 2 prime, 3 prime, 4 prime, 5 prime. 2, what is this linkage? This is 1 prime, 2 prime, 3 prime. So our linkage is 5 prime phosphate group of one nucleotide to 3 prime hydroxyl group of another nucleotide. This is the phosphodiester linkage that we have in all the cases. So let us found that option. 5 prime phosphate group we have in this option A, 5 prime phosphate group. Option B, 3,5 phosphate group, wrong. Fourth says, third option also have 5 prime phosphate group, fine. Fourth option has 3 prime phosphate group, wrong. Now out of option A and C we have to check. So with 5 prime phosphate we join our 3 prime hydroxyl group. It is written as 5 prime hydroxyl. So wrong option. In option C it is written 5 prime phosphate with 3 prime hydroxyl group. So the only option that is correct here is option number C. Fine. Okay. Let us move to the next one. It is a very easy one. It states the torsional angle generated due to the rotation around the bond shown in the following figure is designated as what? So this is here, what bond is this? This particular bond is known as beta glycosidic bond, beta N glycosidic or beta glycosidic bond. Why beta N glycosidic? Because we have this sugar moiety attached with the nitrogen atom of the base pair. That is why it is talking about beta glycosidic N glycosidic bond. So what does it, this rotational angle, this torsional angle rather. So this is a single bond through which the base is attached to the sugar. So any single bond is free to rotate. So what this torsional angle is known as? It is known as the xi angle. Just like in Ramachandran plot we learned about other angles like psi, phi, omega, etc. In case of these polymerized structures, this glycosidic bond, torsional angle around this glycosidic bond is known as xi. Okay, fine. Moving to the next one. It states which type of tautomerization happens in case of adenine and cytosine. So for this to understand, you should remember the structures of adenine and cytosine. Let me show it here. I am showing all the structures. So in the right side we have our adenine and cytosine, and in the left side we have our guanine and thymine. So let us look at adenine and cytosine. What kind of tautomerization is happening here? The H is transferring, this pink colored H is transferring from here to this nitrogen right? So if I mark this as 1, 2, 3. So from number 1 N1, the hydrogen is transferring to N3 and we are having our imine bond. This double bond NH is known as imine bond. NH2, this is known as amine, and this one is double bond NH is known as imine. Similarly for cytosine as well, 1, 2, 3. So N1 to N3, one hydrogen is being transferred and we are getting amine to imine conversion. So for adenosine and cytosine, we have the tautomerization known as amine to imine tautomerism. So this is our correct answer. Although this is not on the question, still just quickly look at the structure for guanine and thymine. What does happen in this case? We do have a NH group here. Now see if I mark this as 1, 2, 3. Now this N1 hydrogen is transferring to the oxygen and forming a OH group. After formation of OH group, what do we call this? This is an enol and initially it was C double bond O. So it was in a keto form. So in case of guanine, the transition or the tautomerism that is happening is a keto-enol tautomerism. Same as for thymine as well. Same transfer, double bond to from keto form to enol form. So for G and T, we have keto-enol tautomerism, and for A and C, we have amino to imine tautomerism. Okay, fine. Let us move to the next one. Okay, this is again another mathematics. It states, suppose a fragment of Z DNA consists of 3 turns. How many base pairs do you expect to be in the fragment? So the only information now we have is that the segment that they have given us, say suppose this is our segment, this has 1, 2, 3 turns. That is all we know. Now they are asking how many base pairs are there. Let us go back to the table, characteristic table for A, B and Z DNA. We are talking about Z DNA. In this particular information, BP per repeat or base pair per turn, it is written already. Then in case of Z DNA, we have 12 base pairs per turn. What is your turn? This one and this one. These 3 are your turns. So in each of this turning, you have 12 base pairs. Altogether, how many turns you have? 3 turns. You already know that. So each of the turns is going to have 12. So total number of base pairs will be how many? 3 into 12. That is 36. So total how many base pairs will you have altogether in case of this particular ZDNA segment is 36. Okay. Let us move to the next one. It says, which of the following represents the correct order of the pitch among the different types of DNA? So, we have our A DNA, B DNA, Z DNA. Sorry, it is actually very much obvious from the table. If we go back to the this particular table, we have our A DNA, B DNA, Z DNA. What is their pitch length? For A it is 2.8, B 3.4 and for Z DNA it is 4.5. But what do you need to remember again? What is the pitch length actually? That is you require to remember. This is the rise per turn. Pitch means rise per turn. That means how much length it has increased for one turn. Okay. So going via the pitch length, for A DNA, this is very short. So it will look something like this. If we compare the A DNA, if the pitch is less, it will look something like this. Compared to that, for B DNA, the pitch is higher. So even if they do have same turns, it will look something like this, because this distance is now higher in case of B DNA, and for Z DNA it is the highest. For Z DNA it will look something like this. Fine, that is your pitch length. Fine. Okay. So our correct option was Z DNA has the highest pitch length, followed by B DNA, followed by A DNA. Okay. This is a very easy question. I'll just quickly go through it. Identify the overall process being depicted in the provided diagram. So what is the process they are showing it? DNA to RNA and from RNA to protein. So DNA to RNA is transcription followed by your translation. Okay, so the overall process is known as central dogma of biology, because in this picture we are showing the whole thing together, not a single process. And in the question as well, it is stated about the depicting image, the whole diagram it is talking about. So the whole process is known as central dogma of biology, where what is X? X is transcription, that is transformation from DNA to RNA. And what is Y? Y is translation, that is the transformation from RNA to your protein. Okay. Moving to this one, it states, identify the correct statements regarding the nucleic acid structures. Correct statement means we have to identify the true ones. Let us read the statements. The first one is, nucleic acids are made up of polymers of 4 different nucleotide residues each. Nucleic acid. So how many? What are the nucleic acid? Deoxyribonucleic acid or DNA and ribonucleic acid. That is your RNA. So both DNA or RNA that are there, both of them have 4 different nucleotides. In this case we have ATGC. In this case we have AGCU. In both of them 4 different nucleotide residues are there. So yes, our first statement is a true one. The next one is, among this, RNA uses AMP, TMP, GMP, CMP. What does AMP means? Adenosine monophosphate. TMP similarly. GMP same. Just in place of adenine it is thymine monophosphate, guanine monophosphate and cytosine monophosphate. Okay. But we all know RNA only uses A, G, C and U. It does not uses thymine. That means TMP will never be present in case of RNA. So this statement is a false one. The third statement says DNA uses DAMP, DCMP, DGMP, DTMP. What does they mean? D means deoxyadenosine monophosphate, and similarly all others. What does DNA uses? A, T, G, C. All of them they use, and they use it, DNA use it in its deoxy form. So yes, the third statement is true again. So according to this question, the true statements are 1 and 3, and we had to identify the correct statements. So the correct option is option D. 1 and 3 statement is true. Fine. Moving to next one. We need to identify the structure of X, Y and Z. So what do we have here? X is a what? You can see in all the options, in case of A, B, C, D, we all know it is adenine. And in bracket, it is written as adenine in all the 4 options. The only difference is, in some options it is written like nucleobase, and some options it is written like nucleoside. Okay. But we all know this is only the base part. So this will not be nucleoside. What is nucleoside? Nucleoside is your sugar part plus your base. Always remember, as I have said, sugar plus base gives rise to nucleoside. Whenever you add another component, that is phosphate, that will lead you to nucleotide. So our X is nothing like that, it is only the base. So our X is adenine and a nucleobase. So this option is wrong, this option is again wrong. So out of A and option C, we are still looking. Okay, what is option number Y? Now the adenine is attached with a sugar moiety here. So sugar plus base will be a nucleoside. So where is the option? You can see. Let me see. Here is the option nucleoside. Yes. 2' deoxyadenosine. Yes, it is 2' deoxy. Why? Because if it is 1' position, it is 2' position. The 2' position, there is no OH group, so it is 2' deoxy, and then adenosine is attached here. Sugar plus base, that means nucleoside. And the third option is, we can see this phosphate group attached here. That means the three of the things are now complete. So altogether they will be known as nucleotide. So the overall, it will be 2' deoxyadenosine, 5' monophosphate. Why so? In 1' position we have our base, 2' is deoxy because we do not have any OH group here. We have our 3' group, we have our 4', and this is 5'. At 5' position there is only one phosphate group attached. That's why it is written as 5' monophosphate. So Z is first of all a nucleotide, then it is 2' deoxyadenosine, 5' monophosphate. So the only option that matches with all these things is option number A. Okay. I think this one is the last question of this session. Let us see. Which are the correct statements regarding exo and endonucleases? So they are actually type of nucleases, right? The first one says they are actually type of ribonucleases, if I have to be specific. So exonucleases, the first statement says, catalyzes the removal of terminal nucleotide 3' and 5' types. Okay, fine. This is absolutely correct. Exonucleases, what do they work on? So we have phosphate backbone both in the exterior and on the some parts in the interior sides as well. These exonucleases always act on this terminal nucleus. These terminal nucleotides, they break. So if this is your 5' end, one second, it is not working. So if it is your 5' end, another will be 3', the next end will be 3' to 5'. So these terminals phosphate groups, they are actually attacked by exonucleases. Both 3' and 5', both the types are actually can be catalyzed in presence of exonucleases. So our first option is a true statement. First statement. The second one says endonucleases catalyze the removal of external phosphodiester bonds. External phosphodiester bonds means that are present on the terminals. But from the name itself, it is very much clear they are called endonucleases because they take on the internal phosphodiester bonds. That is why they are called endo. That means this statement is absolutely a false statement. Moving to next one. Type A endonuclease act on 3' hydroxyl group of the nucleotide with the phosphorus group. Yes, endonucleases have two types. If you classify them, the endo one has two classification. One is known as A, type A, another is known as type B. Type A1 specifically acts on the 3' OH groups of the internal or the internal phosphodiester bonds, and the B type specifically acts upon the 5' hydroxyl groups. Okay, so yes, the third option where it states the A endonuclease act on 3' hydroxyl group is a true statement. And the last one is type B exonuclease act on 5' hydroxyl group of a nucleotide? So first of all, the very first beginning of this sentence is wrong, because exonucleases does not have this classification as A or B. Only endo has this classification of A and B, because exonucleases both targeted 5' and 3'. They do not distinguish or separate them. They target both of them, both 5' and 3'. So they do not have any further classification. So there is nothing called B exonucleases, because there is no such classification that exists. But endo ones have specific targeting, that's why they have been classified. So the very beginning of the sentence was incorrect, that's why the fourth statement was false as well. So in this question we had to identify the correct statement, that is the true ones. So our true ones is 1 and 3. So according to that, our correct option should have been option number C, 1 and 3. Yes, this was the last one. Okay. So, okay, this is it. Thank you all for joining. We'll meet you in the next session. Thank you all. You all can leave now.